Hello students, let's discuss the next process that is called mitotic phase, the cell division phase, the most important phase where cell will divide. After leaving the G2 phase, cell will get enter into M phase or mitotic phase. In process of mitosis, when we talk about the process of mitosis, mitosis is a type of cell division in which one parent cell will be divided into two daughter cells with same number of chromosomes. Means these two cells will be genetically identical with the parent cell. If 46 number of chromosomes are present in parental cell, same amount of number or same numbers of chromosomes will be present in both these daughter cells. This process of mitosis takes place in all somatic or body cells, even in your gametes. When during the process of gametes formation, even over there, this process of mitosis will also be a part of gametes formation. So it means it will take place in all somatic or body cells. When we talk about the process of mitosis, this process of mitosis is a continuous process, but for our convenience, we divide it into 4 phases. Starting with first phase, I have divided it into 2 different forms for my convenience. I mean, like when I learned it, you can take examples of 2 different levels of your educational institutions. I mean, the primary level and matric level. When talk about the primary level, that is the first phase, prophase. There are 4 phases of mitosis, namely P for prophase. When talk about M, that is metaphase. A for anaphase, and 4th is telophase. These are 4 different phases of mitosis. First, we start with prophase and discuss the changes which takes place during this process of prophase. This was G2 phase. After leaving G2 phase, cell was entered into mitotic phase, and the first phase was prophase. When cells enter into first phase, the prophase, the changes which will take place inside the cell, number 1, the nuclear membrane becomes enlarged, thick and prominent. This is the first change. It's prophase. During prophase, nuclear membrane becomes enlarged, thick and prominent. Then what next? The chromatin material, which is actually DNA or histone protein, its coil structure. This DNA material is present in the form of chromatin network. This network starts getting condensed, and when it is condensed, it will be changed into chromosomes. What happens next? Actually, during the process of phosphorylation, when the phosphorylation of these chromatin materials take place, during that process they become shortened, condensed, and they change into special structures called chromosomes. So these are the chromosomes. When we talk about the structure of chromosome, a chromosome is made up of chromatids. It's a chromatid. This chromatid is joined by a special circular structure that is called centromere. So during prophase, these chromosomes are formed. Phosphorylated chromatin material will get condensed, shortened, turned, and will get attached with centromere, which we call chromosomes. Then next, the centrioles. Two centrioles starts reaching to opposite poles. Two centrioles will start to move to opposite poles. These centrioles play a very important role over here. What actually they do? They make tubulin proteins. They give rise spindle fibers. The fibers on which the chromosomes in next stage will arrange themselves on equator. So over here, small thread-like structures starts coming out. When we talk about the centriole, in this centriole, from its backside, esters are originated, the small threads. On front side, the spindle fibers will be generated. So this whole structure where we have a centriole, spindle fibers and esters, it is called mitotic apparatus. This whole structure is called mitotic apparatus, which will play a vital role in cell division. Okay, after or before entering into the next phase, that is called metaphase. When prophase is about to complete, or lateral prophase, this nuclear membrane, which is made up of laminin protein, will also be phosphorylated. When it will be phosphorylated, it means it starts disappearing. So the breakdown of nuclear membrane will get start. And Finally, this membrane will disappear, and when cell enters in next phase, that is called metaphase, what changes we will observe? We observe over there the nuclear membrane completely disappear. The nuclear membrane is phosphorylated and disappear. So what happened in metaphase we will discuss in our next lesson. Till then, stay blessed and stay happy.